In our research, we try to provide better understanding of structure variants, which are genetic changes that are very common in multiple myeloma. They are involved in development of the disease progression and also in treatment resistance. But what we often don't think about that, because they are quite large-scale changes, that not only they are changing the genes, for example, deleting genes within the region, but they are also reshaping how DNA is organized within a cell nucleus. And to access this information, we are not happy only to have genomic data, but we need to use multiomics, so newer technologies and deeper technologies or different molecular level to really understand the consequence of any structure variant. So one of the technologies that we are introducing here is long-read sequencing, where we are able to sequence much longer segments of DNA. So it's something like when we solve a jigsaw puzzle, we can do the picture with very small pieces, or we can have bigger pieces, so we do it with better confidence and much easier, and we know where each segment is going and how it's changing in the patient. At the same time, long-read sequencing technology already offers to understand epigenome, so like modifications of DNA, so we can immediately see what genes are impacted by structure variation. And on the top of it, we are developing techniques where we can also looking at modifications of the proteins that is important in the folding of DNA or how DNA interacts. And if we do that, we really see the complete picture, how structure variation is changing the genome, but also the epigenome and the folding of DNA in myeloma cells. So one point we make in our research is that in omics techniques, we rely on genome assembly, which is like control human genome against we compare our samples. And problem is that if we do so, we kind of miss part of our genome because these references are not perfect. So we are using one of the latest assemblies that was developed by Telomere to Telomere Consortium. So we can access regions that we could not really analyze before. So we are looking at like centromeric regions, telomeric regions, and genome gaps that are very challenging to understand. And one of the change that is related, and we are showing some data about, is 1q gain that is very common in multiple myeloma and is associated with not very good prognosis. And we call it 1q gain because there is additional material on the chromosome 1, but physically, by the principle of chromosomal biology, this material has to be attached somewhere. But because the breakpoint is in the genome gap of previous assemblies, we were not able to see it. So we are trying to resolve this part of the puzzle. So we developed methods where we can fully understand a shape of this abnormality and basically see 1q gain as chromosomal translocation. And we see that where 1q gain of the additional material gets stick to, that there are some epigenomic reprogramming. So I'm looking at if that is also associated with the consequence of the worse prognosis for patients.